think you've thoroughly enjoyed all the presentations, but I think you're going to enjoy this one equally. I had the opportunity to hear Dr. Walter speak in Calgary back in September, and I must say I found it not only informative, but somewhat enjoyable as well. And I did put him on after lunch because he does talk about poop, as I've said that. And, but I think you're going to enjoy today. So I'm going to introduce Dr. Jens Walter. So here we go. And then please don't get up and run away right after we finish the Q&A, because I do want to do the various brief thank yous as well as the exhibit hall prizes. So you're going to want to be here for that. So here we go. She's saying, why are we talking about poop? The gut microbiota and multiple sclerosis is the title, Dr. Jens Walter. As humans, our intestinal tract is colonized by a dense and species-rich community of microorganisms, the gut microbiota. That is of paramount importance to our health. Recent clinical and experimental studies clearly indicate the important role of these communities in the etiology of multiple sclerosis and other autoimmune diseases. In this talk, Dr. Walter will discuss how the gut microbiota is implicated in the pathology of MS and how microbial involvement provides explanations for the impact of the environmental risks of multiple sclerosis. He will further discuss some of the microbiome-targeted treatment options that are currently discussed and researched in this field. Dr. Walter is an associate professor and Campus Alberta Innovation Program Chair for Nutrition, Microbes, and Gastrointestinal Health at the University of Alberta. After receiving his doctoral degree from the University of Hohenheim, how do you say that?
Almost, yeah, Hohenheim.
Almost. That's in Germany. He performed postdoctoral research into genetic and metagenomic approaches to study gut microbial ecology at the University of Otago in New Zealand. In 2006, Dr. Walter accepted a tenure track position at the University of Nebraska-Lincoln to work as a molecular microbial ecologist, and he received tenure in 2012 before moving to the University of Alberta, Canada in 2014. Dr. Walter's research focuses on the investigation of ecological and evolutionary processes that shape host-microbial symbioses, oh my goodness, in the human gut, and the application of these scientific concepts to develop microbiome-targeted nutritional and therapeutic strategies to improve human health. And I'm glad he's going to be able to pronounce these words. Thank you. Yes.
Thank you very much, Angela, for this very nice introduction. It's really complicated what I wrote there, I realized. So it is indeed true that I study poop for most of the time. I study it with quite sophisticated technologies, but at the end it's still the massively parallel sequencing of poop bacteria. Yeah. So I have a disclaimer to make. So I'm not a doctor, and I came into the multiple sclerosis area reasonably late. So by training, I'm a microbiologist with an interest in the ecology and evolution of gut microbes, and that brought me then into nutrition, because I thought it is really important. If you try to understand how your microbiome evolved with us as humans, then this all happened in a nutritional context, and this really then brought me to diseases. And then late in my career, roughly 5 years ago, I became interested in multiple sclerosis. So I don't know as much as the previous speakers about treatments and diseases, but I know a little bit about diet and how it relates to some of these chronic diseases that we heard in the previous talk already. So what I want to do today is I want to give you, of course, a broad, kind of introduction in how the microbial population in our gut are linked to multiple sclerosis, and I would actually argue that this evidence is becoming incredibly strong. And then I'm going to go from there and actually point how this actually then relates to nutrition and potentially some therapies. And we've heard already a little bit by Dr. Wee Young this morning, how important the microbiome is and how people are starting to look at it. But I'm going to talk quite a bit more about this in this presentation. I have a disclosure here, which is that I actually don't have any conflicts as it relates to this talk, as I'm not going to make any specific recommendations towards products. So some of what I'm going to talk about you've already heard in these presentations, which is nice. There is a healthy amount of overlap, but Dr. Young already talked about what is really the cause of multiple sclerosis, and of course the answer to this is that we really don't know what is the exact trigger, but what we do know is that it is a complex disease that has a genetic underpinning and clearly also happens in an environmental context. And Dr. Young already mentioned that we have identified 200 genetic loci that are associated with disease risk, but all of these genetic loci only explain around 40% of the disease risk. So there is a majority of the risk is coming through the environment. And another thing that Dr. Young already mentioned is here the actual findings in monozygotic twins, meaning that we do have a higher incidence of multiple sclerosis in monozygotic twins as compared to dizygotic twins, meaning clearly that the genetic, underpinnings here actually lead to a significant higher incidence of disease. But you can see even in individuals that have exactly the same genome, there is 70% discordance. So what could it be, that actually then, make these individuals differ in getting diseases? And one interesting feature here is that although monozygotic twins have the same actually genome, we do now know from the microbiome field, also the area in which I work, that these monozygotic twins have substantially different populations of microbes in their gut, providing a potential trigger and reason for these actual differences. Another map or figure you've already seen today, just in the previous talk, is the disease incidence, and it is globally. And you've heard a lot about the gradient that happens in disease incidence from the equator to the north, and the potential importance of sunlight and vitamin D. But if I look at a map like this, the thing that I find the most striking is that it really seems that disease risk in multiple sclerosis is to a large degree linked to actually an industrialized lifestyle, or meaning the richer the countries actually get, the more likely is it that a substantial or a higher proportion of the population actually suffers from multiple sclerosis. And as you can see, this is quite nicely visible here, that of course the highest risk is in Europe, in North America, but then you can also see there's a reasonable or quite substantial high risk in Australia, and they really do get enough sun. And then I've just, during the lunch break, actually had an interesting discussion with a lady who pointed out to me, and I actually didn't know this, that Israel has a very high incidence of multiple sclerosis. And again, this is definitely not a country in which you would lack of sunlight, but it is a country with a very, very high standard of living. So I would argue that at least, again, we don't know the exact reasons for these patterns, but I would argue for me the clearest here is really lifestyle, and specifically the actual standard of living. So, and this points to certain environmental factors associated with an industrial lifestyle, and it potentially also could point to the diet, which is one of the unfortunate factors, is actually that the more developed societies get, they often don't eat the healthiest food, which you can actually see on your table at the moment. Yeah.
Thank you. Thank you for pointing that out.
You're welcome. Yeah. I do enjoy that stuff too, so, but I still needed to bring this across. So, if it is the environment, then scientists have already started, and these patterns between Western and non-Western countries, you don't only see this in multiple sclerosis, you see this in others, what we call Western chronic lifestyle diseases. So scientists have argued, could it be that there is a microbial trigger? And then, of course, the most dominant hypothesis here is the hygiene hypothesis, meaning that the more sophisticated or the more modern or the more developed a society gets, of course, the lower is actually the rate of infectious diseases, which you can see here on this graph. They are dropping significantly, and they are very well under control, while then, on the other hand, you have this increase of chronic diseases, chronic lifestyle diseases. Multiple sclerosis is one of it. And I was very fascinated by the previous talk because I was not aware of some of the actually links between some of these chronic diseases and with multiple sclerosis actually pointing to a potential common underpinning. So this is the hygiene hypothesis, meaning that could it be that a lack of exposure to pathogenic organisms, to parasites, and by this then a lack of engagement of the immune system throughout life, then for some reason actually causes an immune response that is detrimental and then increases the diseases. So I'm not sure if the hypothesis, and nobody knows, is right, but the hypothesis and the interest has shifted to some degree in that scientists realize that most of our microbial exposure and our microbial input for our immune system doesn't actually come from pathogenic organisms. The most associations we have with microbes is actually with the bacteria that are just healthy, living on and in our body. And this is what we refer to as the microbiome that Dr. Young already mentioned today. And the densest of these microbial populations is in our gastrointestinal tract. And this is a slide here from 2016. So much of what I'm going to talk about is very recent, but there's quite a substantial interest now to test the hypothesis. Could it be that multiple sclerosis actually has an association with the gut microbiome, and that this is actually then contributing to the development of the disease? So before I go into specific, I would like to give you an introduction about what we talk about if we talk about the gut microbiota or the gut microbiome, and also want to point out really how significant of a player these microbial populations are to our biology. So you can see here from this graph, this is the gastrointestinal tract, and all of these areas are colonized by microbes. But if you look down the intestinal tract, then you have very few microbes in the stomach. And then the further down you go in the small intestine, and you move slowly into the ileum, which is the end of the small intestine, you already have 10 to the 8 microbial cells. And then in the colon, we end up to get 10 to the 11 microbial cells per milliliter of gut content, or in other words, 100 trillion microbial cells, most of which are bacteria. Just to put this in perspective, as you sit here at the moment, your body has as many microbial cells as it has human cells. So if you considered yourself primarily human, biologically, you're only around 50% human. Yeah? The other 50% are composed of microbes. And if you view this from a genetic component, it's actually even more extreme, because our human genome only has around 23,000 genes, while this microbiome, so meaning the complete array of genes that are associated with these microbes, end up to be more than 1 million genes. So as genetically, you're by far more microbe than you're actually human. Yeah? And obviously, these genes do something. These genes allow these microbes to colonize us, to be metabolically active, to interact with our immune system. They help us digest food. So many actually functions are, that we obtain basically many functions from these microbes. And I like to refer to this interrelationship as a co-evolved symbiosis. So basically that these microbes, and this is one of the really big take-home messages today, these microbes are not really just there. They are essential for our entire biology. They are essential for our development, and they were important over millions of years while our bodies, while our functions, while our immune system actually evolved and have shaped us through their actually benefits that they have provided. There are really many, many examples, but the most important contribution that microbes make is that they help us to extract nutrients from the diet, which is actually probably something that was very, very important throughout human evolution. It is probably not important anymore. In fact, in the field, we actually consider this detrimental now because we have an obesity epidemic in the Western world, and the gut microbiome is implicated in contributing to this. So a trait that probably was very beneficial over evolutionary time due to the environmental changes is now detrimental. What another very important benefit that we receive from these microbes is resistance to infections. Some of you might have heard about Clostridium difficile diarrhea, which is becoming quite a serious threat. So Clostridium difficile diarrhea is a pathogen. Most of us will probably have it in our gut, but it doesn't matter in healthy individuals. And the reason why it doesn't matter is because we have a healthy gut microbiome that keeps that pathogen at check. It competes with it, and the pathogen can't really grow. However, if in certain circumstances, specifically by chronic antibiotic treatment, this you disrupt your microbiome, that pathogen can actually bloom to higher numbers and then causes severe illness. And I'm going to talk a little bit about this later. And then finally, and probably the most relevant to diseases like multiple sclerosis, is that these microbial populations are absolutely central in the development of the immune system. Also meaning this, during the early colonization in life, our immune cells actually sense, interact with bacteria and with microbes, and by doing this, they actually are influenced in the maturation. And this is probably one of the key points while the microbes contribute to actually host performance, but also why they are linked to diseases. So just to summarize it, we have a symbiotic relationship in which these these very dense and cell-rich populations contribute to our biology. But then on the other hand, and this is why the gut microbiome field is receiving such a lot of interest, on the other hand, these microbial populations have also clearly been linked to chronic diseases such as heart disease, colon cancer, obesity, diabetes, and autoimmune diseases such as multiple sclerosis. And I'm actually missing one part here. These microbial populations have also been linked to mental disorders such as anxiety and depression. So I was very interested by the previous speakers' information that multiple sclerosis is linked. And I don't want to talk too much about anxiety again, but the gut microbiome has been shown to be actually also influencing the brain and actually linked to anxiety. And as we also heard in the previous talk, is that some of these morbidities are actually associated with each other, and they have a common underpinning in inflammation. So clearly you could now say, so why is this scientist here telling me first that this is a symbiotic, interrelationship that is mutually beneficial, but then on the other hand, we have this link to these diseases. And for me, the key, again, and I've shown this slide already, is the fact that these diseases have really not been that important more than 70, 80, 90 years ago. And we do see this increase actually associated with a modern lifestyle. Putting forward the idea, could it be that due to the lifestyle, we actually disrupt this microbiome? Due to this, we don't get the benefits from these microbes anymore, and then are predisposed to diseases such as multiple sclerosis. And this is a hypothesis that has been brought forward around 10 years ago by scientists such as Marty Blaser and Stanley Falco. They framed the hypothesis of a disappearing microbiome. So in other words, is that modern lifestyle is actually depleting our microbes. And by this, we are not receiving the benefits anymore that we have actually, received from these microbes throughout evolution. And what is nice now is that we actually, in fact, have the data now to prove this hypothesis. We have a depleted microbiome, and we do know this from many studies that compared the microbiome in modern societies, such as the US, for example, in Europe, with the microbiome of individuals that live a non-industrialized lifestyle. And these are probably two of the publications that studied the most extreme of the human populations. And I had the privilege to be involved in both of these publications. So the first publication was led by Maria Gloria Dominguez-Bello, and it studied the gut microbiome in a very fascinating group of individuals. It was a study that looked into the guts of uncontacted Amerindians. So this is really as far away as you can get from an industrialized lifestyle. So these are individuals that live in the Venezuelan rainforest. They have minimal contact to the modern world, and you need to basically go there by boat and by expedition to actually, and they're protected by the Venezuelan government, so to actually don't have extensive, and you can see one of these pictures here. The second study was led by myself, and it was a study that studied two tribes in rural Papua New Guinea. Papua New Guinea is one of the most diverse countries on earth. It's next to Indonesia, with which it shares a border, but the most important thing is that due to its hilly terrain, the actual diversity of individuals is just, and cultures is actually mind-staggering. So 1/3 of the world's languages is in Papua New Guinea, so they speak 700 different languages. And because of the terrain and about the isolation, many of these tribes also still live a very, very rural and non-industrial lifestyle. They are not uncontacted like these Amerindians. And again, these Amerindians are also contacted, but only by doctors and by expeditions. These Papua New Guineans have more contact, but they still live a non-industrialized lifestyle. So this is a picture that actually, this is Maria Gloria Dominguez, and I acknowledge her contribution here. She sent me that picture. You can see these are the Yanomami, the Amerindian tribe in the Venezuelan rainforest. You can see you can't go there by car. And this is Maria Gloria on one of these exhibitions going there by boat. And one of the anecdotes I always like to share is, so Maria Gloria told me that you go on an exhibition and these individuals have really not had a lot of interactions, with white-looking Europeans. And then one of the first things you're asking them if you can get their poop sample, yeah? But she says actually what they do is they basically laugh at you. And they think it is incredibly funny, but they are actually willing to collaborate, yeah? And so this is a major achievement, really, to get these samples, and then in collaboration with Jose Clemente, Maria analyzed the gut microbiome, and it's not that important to read this, but this is a diversity graph, and the way how you read this is this green speck here on the left are the Yanomami, and it basically shows that they have the most diverse microbiome that we have seen so far, meaning non-contacted lifestyle means they have a lot more going on in their gut than we in the West. And here the yellow is actually the US controls. As you can see, there are other non-industrialized populations which are slightly less diverse, but then the worst is, of course, the industrialized nation control. In Papua New Guinea, this is the study that we did, and my collaborator Andrew Greenhill was in charge. I've actually never been to Papua New Guinea, but he organized the fecal samples. We paid $5,000 to ship poop samples to the States in these days on liquid nitrogen to be able to study their microbiome. And same story here. If you look into Papua New Guineans' guts and you compare this with American, a significant higher amount of diversity. And this is the heat map of all the bacteria that we find in these two tribes in Papua New Guinea. And you can see there's quite a bit of overlap in the microbiome, showing, we are all human, and due to our coevolution with these microbes, we actually assemble the same bacteria. But then what is also striking is that you have this entire chunk here of microbes that are completely absent in the US people. Around 50 species were not detected, really supporting this idea, that yes, the gut microbiome in the West is depleted. And on the other hand, what you can see is we only detected 4 species that are present in the Americans that we couldn't find in the Papua New Guineans. So that means our microbiome is depleted, but we don't really get anything in return. So supporting, again, the disappearing gut microbiota, hypothesis. Yeah. So I would argue that this really sheds a light that could it be that it's not so much the parasite, but could it be that it is the microbes that are missing that actually lead to these, increase in diseases. And one disclaimer I have to make, which is very important, is that on average, Papua New Guineans and Amerindians are not healthier than, individuals in, the US or in Canada. Their life expectancy is significantly shorter, and they do suffer from many of these infectious diseases, specifically in Papua New Guinea. The data is actually clear that they have a very, very high amount of infection, high amount of infant mortality. But on the other hand, they do have a significant lower amount of chronic diseases, such as multiple sclerosis. We actually tried to find information about multiple sclerosis in Papua New Guinea, and we found one single case in the literature. So it's really not a very common disease there. So the idea is not to really, advocate their lifestyle. It is more to try and understand, why is it, or what is actually lacking in our life, that we have these increased incidences, and then due to understanding, potentially actually try to fix it. Yeah. So what is it that depletes the microbiome? It's definitely the context in which the microbiome functions is completely different in the industrialized world. It has fundamentally changed compared to the conditions in which this interrelationship has evolved, and it is completely different than a non-industrialized lifestyle. So what is different? Of course, we have high amount of antibiotics that we consume from early in life. We have small households under very hygiene conditions, while Papua New Guineans, for example, often live with actually animals in the same households. We have hospital deliveries, cesarean sections, meaning a large proportion of children get now brought to life by a cesarean sections, which means that the actual natural transmission of microbes very early in life is actually disrupted, because if you cut open a belly under completely sterile conditions, these infants do not receive the early microbial kind of inoculum that you would get in comparison to a vaginal birth. And then, of course, we do have sanitations, which again comes with all kind of advantages, much less infections, much less pathogen exposure, but also could it be that we actually, with all of these lifestyle factors, actually induce hurdles to symbiont transmission? So meaning we just don't acquire the symbionts anymore as easy as in non-industrialized populations. Then probably just as important is the diet, which has substantially changed in the last 100 years due to industrialization. And one of the dietary factors I'm personally most interested in is dietary fiber. So throughout our evolution, we have consumed 150 grams of dietary fiber every day. And today, we are lucky if we are actually getting 15 to 20 grams of dietary fiber. And even if you follow a very healthy diet, it is very hard to actually boost fiber levels. And I'm going to come back to this. Then we do things like formula feeding. Human breast milk is full of nutrients for the microbes actually in the infants, which are absent in formula, which is one reason why the microbiome early in life is hypothesized to be disrupted. And then what is probably also absolutely central are the high amounts of simple sugars and saturated fats that are in modern diet. Look on your table if you don't believe me. Yeah? Sorry.
I didn't bake it.
It's all good. So, and again, sugars and saturated fat, of course, there's a lot of information in nutrition and in nutrition research that they are detrimental, but they also have been shown in many experiments to disrupt the microbiome and cause chronic disease due to this disruption. So for me, also what this factors here cause is that we have an insufficient dietary support of our gut microbiota in modern life. So essentially, we are starving our own microbial partners, and this then has detrimental outcomes, meaning these partners, if I get starved, I'm also not willing to provide the benefits anymore that I have evolved to. So we are losing these benefits. And I do really like the term here of the old friends hypothesis, meaning that due to lifestyle changes, we are actually losing, old friends, meaning, partners that we've actually evolved with over millions of years, or at least we are losing the benefits that we have received from them. Yeah. So importance of diet is easy to show. This is pictures here provided by my collaborator Andrew Greenhill from Papua New Guinea. So this is the diet that is consumed in these tribes that we studied, and as you can see, it's a diet very, very high in root vegetables and leafy vegetables, substantially amounts of dietary fiber, and much, much higher than in the West. Then a key study here, this is a study from Justin Sonnenberg's group at Stanford University. So what this group did is it fed a diet that is basically devoid of what they refer to as microbiota accessible carbohydrates, which is in other words dietary fiber, also meaning carbohydrates that survive digestion, that reach the colon, that then feed the microbes. And they did a very elegant experiment in which they put mice on either a diet with dietary fiber, and you can see all of this is the microbiome, and over the generation you can see most of this microbiome is actually maintained, while this is the mouse group in which received a diet that is devoid of dietary fiber, meaning a diet similar to Western food. And as you can see, within just 4 generations, they have lost 70% of their actually microbiome. Yeah, so providing clear evidence that this depletion that we see in the West might in fact be due to us not providing, substrates, growth substrates for these actually microbes. Yeah. But it is not only the composition, we are not only losing bacteria, but they also change functionally, which is relevant. This is more work by Justin Sonnenberg's group. And this here is a micrograph, which is probably my favorite picture from the microbiome field. What this micrograph here shows is the epithelium in the colon of a mouse. So these blue areas here are the mouse cells. And then you can see that the epithelium actually has cells here, goblet cells that produce mucus. And this mucus layer is shown here in green. And then we have these trillions of bacteria from which I just told you, which are stained here by different dyes that are in the lumen. And one very important thing here, which is central to our peaceful interrelationship with these microbes, is that the mucus that is produced is actually keeping the microbes away from the epithelium. And you can very nicely see this, that you see these microbes are colored here, and you can see virtually no bacteria within this mucus layer, so meaning it provides a healthy barrier between our immune cells and actually the microbes that we have in the gut. And what Dr. Sonnenberg's group now showed very elegantly is, again, if you give a diet that is deprived of dietary fiber, what these microbes do, because they have nothing to eat anymore, they basically just start nibbling on the mucus layer, and what you get is a significant lower or thinner actually mucus layer. So what it means is if you starve your microbes, they basically turn on against yourself, start to degrade the mucus layer, and by this come closer to the epithelium, closer to the immune cells, and actually by this driving an inflammatory response. And this is caused by low fiber, but it's not only caused by low fiber, there's evidence that saturated fat does it, and very interesting, a paper that was published a few years ago, emulsifiers, which are very, very common in Western foods, and I'm not pointing to the table anymore because I might get into trouble. Yeah. But many actually modern, processed foods contain these emulsifiers, and again, emulsifiers have been shown to degrade the mucus layer in our guts. Yeah. Emulsifiers are chemicals that are added to modern food to actually keep fat in emulsion. Also meaning if you want to, for example, generate a margarine, which is very, very, rich in fat, and it's a mixture of water and fat and water doesn't really mix, so you would actually get a separation. So you would have basically a layer of water on your margarine. To avoid this, emulsifiers are actually added, and then they keep the fat and the water within a mixture which is called an emulsion. that are emulsifiers. So many, many modern products actually contain this. And because of the chemical structure, these emulsifiers also disrupt the layer of mucus that we have in the gut. Yeah. So if you look at this, what we basically get, and this is a paper that my own crew published with Frederick Beckett, you can really see that the Western diet with several of the components like a low fiber content, high saturated fat, emulsifiers, basically essentially, disrupts the homeostasis or the healthy relationship that we have with these microbes in the gut. The microbes move much closer to the epithelium and you actually get an inflammation. Essentially, a central factor here is actually that these dietary fibers get fermented in the gut by microbes into short-chain fatty acids. And these short-chain fatty acids have central roles in educating the immune system. And we've heard a lot about bad and good immune cells, or at least cells that act, aberrant or not beneficial under certain conditions. Short-chain fatty acids have in fact shown to induce actually immune reactions that are more beneficial, that are more anti-inflammatory. And without dietary fiber, you have a significant lower amount of these immune cells. There's also fascinating new research is actually that these short-chain fatty acids remove oxygen from the epithelium and keep actually our gut deprived of oxygen, which is again important for bacteria not to grow at these epithelium. So it's a very strong line of research really saying that our modern diet essentially disrupts the healthy interrelationship that we maintain with the microbiome at the epithelial border. Yeah. And we've heard already quite a few times actually today, Dr. Young mentioned it, this idea of a barrier dysfunction or a leaky gut that has been shown to be associated with chronic diseases, and it also has been shown to be higher in diseases such as multiple sclerosis, and there is interest now to study that more mechanistically. Again, evidence that it's actually true what I tell you. This is another fascinating paper by the Sonnenberg group looking into the actual microbiome of humans that live in Tanzania and that follow a hunter-gatherer lifestyle. So this is the lifestyle that we followed over most of our evolution. So these are the Hadza hunter-gatherers. And these scientists looked into their gut, and this is really fascinating. So they basically sequenced their entire poop again. And by sequencing and then analyzing this bioinformatically, they could identify the array of enzymes that are produced by these microbes. And what they could show is that the gut of these Hadza hunter-gatherers is, as the microbes have a very, very high ratio of enzymes that are targeted toward plant carbohydrates because their diet is so rich in dietary fiber. While if you look into the American controls that they had in this study, they could actually see that most of the enzymes in these microbiomes was actually targeted against the mucus layer. As again showing that in a non-industrialized mindset, the microbes are basically specialized and focused on the diet, on the dietary fiber, on the plants, while our microbes are happily nibbling away on our mucus layer. And here this is a paper by V. Young, which you just heard this morning, showing or discussing the importance of this intestinal barrier for the pathophysiology of multiple sclerosis. So I want to shift gears a little bit and show you something about the actual evidence for why we think the microbiome is associated with multiple sclerosis. So first, and this is coincidental evidence because it's not direct, is that environmental factors that are linked to MS, such as antibiotics, C-sections, high sugar, high fat, are also disrupting the microbiome. So this here are papers showing that antibiotic use early in life, cesarean delivery, and also formula feeding are also all associated with an increased risk of multiple sclerosis. And all of these are factors from which we know that they actually also disrupt the gut microbiome. Clearly, this is not direct evidence, but I do find this is really interesting data because first, it allows us to potentially change clinical and nutritional practices, but it also provides a potential mechanistic explanation for why these factors are actually potentially linked to multiple sclerosis. Then the strongest evidence for a role of the microbiome in MS is work with animal models. So there are probably several dozens of animal models now that due to some changes in the immune system actually develop diseases that are at least similar to multiple sclerosis, such as experimental EAE, also experimental encephalomyelitis. And this here is just a list of these actual mouse models, and if you that actually show that if you raise them without microbes or if you give them antibiotics, the disease actually substantially changes. And this here is data that Dr. Jong already showed this morning. So this is the mouse model developed by Hartmut Weckerle's group in Germany at the Max Planck Institute in Martinsried, which is a mouse model that is a transgenic mouse model that expresses a specific T-cell receptor. And these mice develop spontaneous relapsing and remitting paralysis that resembles human MS. And you can see if you keep these mice under normal conditions with gut bacteria, you get around 80% decrease. But if you raise them under a completely germ-free environment, they actually don't really get any disease. They are completely protected. And then when you colonize these mice, they actually develop a disease. And this was really, this was published in a Nature, so a very high impact journal, really showing that just a healthy microbiome to some interactions with actually immune cells can introduce or is completely responsible for the disease in this actual animal model. So this is the strongest evidence that we have. And then finally, there are alterations in the gut microbiota that are associated with human multiple sclerosis. And this is a recent review showing about all the actual changes in the microbiome that have been shown in patients with multiple sclerosis. Also meaning that if you compare patients with healthy controls, you see changes in the microbial populations. Clearly, this doesn't necessarily provide evidence that these microbial populations contribute to disease, because it could just be the result of the disease. However, and this is also something that Dr. Jong already shown, there are now two publications that have implanted poop from multiple sclerosis patients and from healthy controls into two of these mouse models of multiple sclerosis. And what they found is that there is a higher incidence of disease if you implant the poop of multiple sclerosis patients, showing that in fact there is something in the microbiome of individuals suffering from multiple sclerosis that can trigger disease in these predisposed actually hosts. And this again is a strong evidence that there is in fact a causal interrelationship between the microbiome and disease in multiple sclerosis. So I only have one slide because the previous speakers knew a lot more about the actually mechanisms, but this here is a slide from Hartmut Weckerle, who is from the Max Planck Institute in Munich, and this here is a model by which he thinks actually the microbes contribute to multiple sclerosis. So the model is that you have generations of autoimmune T cells in the thymus, which is a normal process. These T cells then get activated in the gut, also in the lymphoid tissue, and there is evidence now that this happens either due to gut bacteria or to lack of certain metabolites from gut bacteria, and then these autoimmune and now activated immune cells then actually basically migrate to the brain, and you've seen the slide from Dr. Jong today. They are then translocate through the gut-brain barrier into the brain and then cause disease. So clearly this, and Dr. Weckerle thinks that this is a central actually immune activation that happens in the gastrointestinal tract to interactions with the microbiome. And again, I'd want to throw out this term here of the leaky gut, because the hypothesis is that the more bacteria and bacterial products can actually translocate through the barrier, which I just showed you is maintained between bacteria and immune cells. The more bacteria can travel through this, also the more we have a leaky gut, the more likely is that these pathologies develop and potentially also that they are actually maintained in these individuals. So this all provides a rationale that the microbiome and metabolic activities of the microbiome as related to diet play a role in MS. So clearly this does open up, possibilities to target now the microbiome for therapeutic implications. And I have to, again, do a disclaimer here. This is a research field which is essentially only a few years old. So there are not any, treatments comparable to the ones that we've actually discussed during this conference yet that actually target the microbiome. However, what I do think, there are implications for human nutrition and how it relates to the gut microbiome, which I think can provide helpful guidelines for patients with MS. So I want to shortly discuss some of these developmental strategies and then finish my presentations actually talking a little bit about diet and how it relates to the gut microbiome. So the gut microbiome can be influenced by different strategies. We can, for example, add bacteriophages to the microbiome. Bacteriophages are viruses that infect bacteria. So we can target bacteria with bacteriophages. This is an experimental treatment. It is heavily studied, but there is nothing yet on the market. And we already talked about antibiotics and some success stories, and I'm talking later a little bit about Dr. Young's research here. Then we have probiotics, which are live bacteria, and I will talk a little bit about probiotics and what are available. And just a disclaimer here, there is currently no probiotic that is recommended for, despite the promises that some of the early research show, there is not yet anything on the market that is actually recommended for multiple sclerosis patients, but that might change very soon. And we have a strategy referred to as prebiotics. Prebiotics are essentially dietary fiber. So you can get supplements of carbohydrates that are non-digestible that then beneficially influence the microbiome, but it is essentially a dietary fiber. And there's quite a bit of interest in fecal microbiota transplantation, and finally diet. So first, probiotic, and I get this question all the time. So probiotics are live bacteria. You can buy them in healthcare stores. You can get them as supplements, as pills. You can get them in food, yogurts, salad dressings. Probiotics are added to a lot of products and can be consumed. These microbes survive actually the stomach, and they do end up in your gastrointestinal tract. This is very well studied, at least for good probiotic products that happen, and you can find them in the poop samples alive. So they basically travel through your gastrointestinal tract. Important thing here to consider is they are not changing the composition of your gut microbiome. So they are basically just traveling through, at least the products that are currently on the market. As I said, there are no probiotic products that are recommended for MS, but if you're interested, I actually just found this site, which is actually a Canadian site, which provides information about probiotic products for which there is clinical evidence for different, for example, mood and depression, and for conditions such as IBD, also irritable bowel syndrome, which might also be associated with multiple sclerosis. So if you go to this site, you can actually find the products that are currently recommended, also some which are actually approved by Health Canada for these conditions. Again, no product, yes, for multiple sclerosis directly. Then what received tremendous attention in recent years is fecal microbiota transplantation. So fecal microbiota transplantation is exactly what it says. So if we assume that there is something different in the microbiome of patients, and that this contributes to the disease, that stimulates, interacts with the immune cells in a detrimental way? Could it be possible to replace the microbiome with a healthy microbiome? So this is exactly what fecal microbial transplantation means. It means that a healthy poop sample will be introduced into the gut of a patient. And you can go both routes, through orally or anally, and then infuse this. So it is good that this talk is not during lunch, yeah? So fecal microbial transplantation has tremendous success rates in Clostridium difficile diarrhea, which is the pathogen that I've just mentioned at the beginning of the talk, with around 95% cure rates. In multiple sclerosis, there are some anecdotal evidence that it actually does something, and there are clinical trials that you can find in the internet that are ongoing, but currently, again, there is no recommendation that fecal microbial transplantation is currently recommended. But again, this is a research that is currently ongoing. Then you've already heard about the trial of V. Young of minocycline, which I find interesting. Dr. Young mentioned that this is an antibiotic, but this also has substantial interactions with immune cells and immunomodulatory effects. But it is, after all, still an antibiotic, which is likely to also influence the microbial population, specifically the bacterial populations in the gut. So you've heard already about, and you heard it from the actual investigator, that there is a benefit in this, and clearly seeing that an antibiotic has benefits is definitely something that should be further researched. I do share Dr. Young's opinions on antibiotic treatment, though. There was a question, what is about harsh, treatments of antibiotics? They do disrupt the healthy microbiome also. They do lead to antibiotic resistance in bacteria. So this is clearly something that is not generally recommended in general, and really just applies to specific compounds that then have to be researched. So this brings me then to what is probably the safest advice that I can give, and I do think there is a tremendous promise in actually applying the right kind of nutrition to first prevent diseases such as multiple sclerosis, but also to manage diseases like multiple sclerosis. So what is, for everything what I've told you today, what implication does this actually have for nutrition in multiple sclerosis patients? And to come back here to Dr. Weckerle's slide, clearly if these immune cells are actually activated in the gut through bacterial interaction, is it possible to change this environment due to diet by providing, for example, dietary fibers to have microbes ferment them to metabolites that have then immunomodulatory activities, and then prevent certain aggravated immune responses in the gut, and by this having benefit in multiple sclerosis. So this is again the slide from our review. So in contrast to unhealthy diet, if a diet very rich in dietary fiber, plants, vegetables, fruits, very important, it was also already mentioned by Dr. Young, diet high in omega-3 fatty acid like fish, a diet low in salt. This has been shown, and there's quite strong evidence that it in fact protects the intestinal barrier, and then protects actually from underlying inflammatory responses in the epithelium. And I'm just showing you a few examples here, not related to multiple sclerosis, but these are two publications from this year showing that dietary fiber can in fact re-establish this healthy barrier at the epithelium, and by doing this induce beneficial immune responses in the gut, and protect then from other inflammatory conditions such as inflammatory bowel disease. This is a study by myself in collaboration with Gurumurthy Krishnamurthy's group at the Max Planck Institute. This is, again, an animal model of multiple sclerosis, and the Guru's group gave these mice a diet very rich in dietary fiber, and as you can see here, there was a substantial improvement in actually the disease, clearly showing the dietary fiber can actually have a benefit in a model such as this. There's a substantial amount of interest in research in compounds that actually signal through the aryl hydrocarbon receptor. I really do not want to go through these processes here because it's highly technical, but at the end, these are compounds which are very common in certain vegetables, specifically broccoli, which then actually sends through these receptors, inducing immune responses that are actually considered beneficial, which includes immune responses linked to neuroinflammation. Yeah. And then finally, you can see supplements such as omega-3 fatty acids have been shown to protect the microbiome and prevent inflammation. We've already heard quite a bit of vitamin D, and then sodium was also already mentioned, which has been shown to induce detrimental immune responses. So definitely a diet that is low in sodium is probably likely beneficial. And Dr. Young already also mentioned there is a surprisingly low amount of strong nutritional research in multiple sclerosis patients to really make definite recommendations in this area. And as you can see, this is actually from the internet page of the MS Society, which shows that there is, indirect or coincidental evidence that diet is important, but there you can't make any strong, recommendation. Nevertheless, the diets that have been researched do actually show very interesting patterns and, are consistent in actually many of the compounds that they recommend. And so I'm going to go through this. And what I actually just learned by looking at the history, which is out there in the other room, the Multiple Sclerosis Society, I didn't actually know that the first grant that was awarded was actually on the Schwank diet more than 50 years ago. So the first, yeah, that was the German pronunciation, sorry, yeah.
We like it.
So showing that there are also interest is definitely there to use diet, but despite more than 50 years of research now, there's still no absolute definite proof. However, many of these diets are consistent in that they actually recommended high amounts of dietary fiber, fruits, vegetables, nuts, the replacement of red meats by white meats, specifically fish, the consumption, the replacement of animal fat with healthy fats from fish such that contain high amounts of omega-3 fatty acids, and definitely also a reduction of salt. And one of the dietary advice that and I don't only use this if I speak in front of multiple sclerosis patients, but I also use this in general, dietary recommendation that I find very strong is the healthy eating plate from the Harvard Medical School, which you can see here is actually quite easy to follow by providing, healthy oils in the diet, definitely a high amount of vegetables. So the lunch was actually very good. I have to say that, yeah. Also by providing chicken instead of actually red meat, plenty of fruits. One thing that I was not aware of when I saw this for the first time around 5 or 6 years ago, that actually fruit juice is not considered to be very healthy because it is too high in sugar and it doesn't really contain the dietary fiber anymore. So definitely reduce the amount of fruit juice. Then for baked goods, so definitely our starchy goods, go from refined products to whole grain products, which again contain dietary fiber, but also other, phytochemicals. And then we already mentioned definitely fish, but also nuts are considered. I always get asked why I am showing this healthy eating plate instead of the Canada food guide. My nutrition colleagues always challenge me here. The Canada food guide is actually quite similar, but it differs mainly actually in the recommendations of dairy foods. And I do have to say this is a controversial topic, and I don't want to have too many opinions. I would also say that dairy foods is a very, very broad range of products. I would argue that actually certain low-fat yogurts are probably quite healthy, while a 60% fatty cheese, as good as it is probably not as healthy. And this is the reason. So I'm again, if gut feeling tells me that yes, fermented foods, specifically yogurt, and then cheese in moderation. Important thing is also to consider that everything in moderation all of this should actually be consumed in moderation. Yeah. So. There are some, again, there are some clinical trials, and I show some of them, but there's definitely much more research needed. So there's not the strongest clinical evidence when it comes to these diets, but there are some, encouraging anecdotal evidence, for example, by this individual here, Dr. Stenzig, which is featured on this Forks for Knives internet site, which is an internet site which basically promotes a full whole plant diet, which definitely has a lot of healthy aspects. And this individual here lives with multiple sclerosis for 20 years without any current signs of disease, actually. And then the little bit of clinical research that has been done in controlled studies, there is a pilot study here that compares the immunological effects of diet on the microbiome, and this here is a whole plant diet in comparison to a normal diet, and they do show, and it's a very, very small diet, but they do in fact show that there is a lower degree of relapses with the whole plant diet. And finally, there is this systematic review here on clinical studies which looked at PUFAs, which are polyunsaturated fatty acids, as common in fish. And you can also get them in supplements. And if you look into these studies, most of them actually show a benefit. And then there are quite a few interventions now on vitamin D. So again, much more research is needed, but there is definitely some promising data with polyunsaturated fatty acids, which support also the consumption of fish. And unfortunately, there's currently no clinical trials, at least not that I am aware, on supplements such as dietary fiber, also that you actually consume dietary fiber in pill form, which is something that my group is actually quite interested in studying now. So with this, I would like to conclude. I hope I convinced all of you that it is worth to actually look into your gut and consider that when you eat, you're not only feeding yourself, but you also feed, trillions of microbes that are important for your body to function correct. Yeah. The research in this area is clearly very, very active to try and identify causative and protective agents within these microbial communities and to develop therapeutic and nutritional microbiome-targeted strategies, and I'm involved in some of this research. Yeah. Diet impacts the microbiota diversity. It protects the composition and function of these microbial population, and it definitely improves the interrelationship of microbes with the immune system and with the physiology in the gut. And this applies to multiple sclerosis, such as it applies to healthy individuals. Yeah. Western diet induces, and this is clear now, it includes what we refer to as a dysbiosis, meaning it is an aberrant microbiome, and specifically also it disrupts the barrier, causes a leaky gut, and through this systemic inflammation, which is likely important in these diseases. Yeah. In contrast, a healthy diet supports the gut microbiota and has potential to prevent chronic inflammatory and autoimmune diseases, and it clearly could serve as a complementary therapy in diseases such as multiple sclerosis. And clearly, although this is a topic for of interest for the MS community, and I just learned for 70 years now, there is very few well-designed randomly controlled intervention trials exist, and this really is a need and potentially also something that probably the MS society could probably consider, having a focus on in their funding priorities, and I'm hugely biased by saying that. Yeah. So, what I do feel very strong about, and this is the nice thing here is, it's not so much about multiple sclerosis here. This here is a general recommendation that I really believe in. And if we now, in the times where we have chronic diseases such as obesity, affecting 50–60% of individuals, and other chronic diseases, I think it is probably fair to say, and the evidence is starting to be very strong, that following this healthy eating plate is probably very beneficial. And what I actually realized is so Dr. Young today said in his talk, that you should probably just follow, what feels good for you. So I'm not sure if I 100% agree with that, because it does take some effort, to follow a diet like this. However, I can also say it is actually possible without a substantial, reduction in quality. And I actually just did the experiment yesterday evening here in the restaurant, where I had a big salad and wild salmon, which is full of omega-3 acids, with vegetables and some balsamic and olive oil, balsamic vinegar. And I do have to say it was the dish I anyway felt most like, but it was not tough to actually eat this. And this would follow very much the healthy eating plates. Of course, there's some issue also that a diet like this is not necessarily cheap, also, so there is definitely some complications here. But it's worth having to think about it, and if it's actually done well, and if one puts some effort into this, it is not necessarily a diet that is, not of high quality and also not enjoyable. Yeah. With this, I would like to acknowledge my postdoc, Elisa Perez-Munoz, who is doing most of my research, Ines Martinez, who is the postdoc who was responsible for the research in the Papua New Guineans, Dale Archer, which is my new master's student, my collaborator Chris Power from the University of Alberta, my collaborators in Germany, Gurumutha Krishnamurthy and Kerstin Behrer, and finally my collaborator in Australia, which is also often in Papua New Guinea, for providing us with these samples. And with this, I would like to thank you for your attention and encourage you to feed your gut microbes. Thank you.
We got quite a few questions here, so let's get through them, because I know we're already at the end of our day, but I want to cover off as many as possible. So is goat milk inflammatory like dairy? Quickly, you didn't really have much to say about that.
I don't really. So the question was, is goat milk less inflammatory than dairy? I just said I am divided about dairy to begin with. It's controversial in the nutrition field. Some societies recommend dairy, some don't, as you could see. So there's no consensus, and there's definitely no consensus on goat milk, I would think. Yeah.
Kelp or seaweed, is it good form of iodine for MS?
I don't know anything about iodine, but seaweed is rich in dietary fiber, and I like dietary fiber. Yeah.
And you've already spoken about probiotics, so I appreciate that you've answered that question. I assume that was correct. The keto diet.
So the keto diet is essentially a high-fat, very low-carbohydrate diet. So the keto diet shows benefits in certain medical conditions, and it is excellent if you want to lose weight. As a gut microbiome, we think it's almost the worst-case scenario, because you have the high fat and the high protein is actually inducing exactly what I've shown you with detrimental metabolic outcomes in the gut. You would probably disrupt the mucus layer. So probably in the long term, it's probably not beneficial. And in terms of multiple sclerosis, the disease is the research hasn't been done. But from everything how I see the gut microbiome in its relation to health, it's probably not beneficial.
A few questions about gluten or wheat. Is it inflammatory in this case?
I follow the consensus here that gluten is only a problem for individuals with celiac disease and gluten intolerance.
I really like this question. Can a better diet create a healthy microbiome, but in elderly people? Is it too late sometimes to turn back time?
So that's a very good question for many reasons. So I didn't talk about this, but the microbiome in elderly people is actually even more depleted than our microbiome in general. So it actually, the diversity reduces with age, and this is linked to then inflammation. And there are efforts of different research groups to actually boost this diversity through probiotics, through dietary fiber, through a healthy diet. So, and to what degree this can actually be done, I would argue isn't really studied or understood yet. But clearly the recommendations in terms of a healthy diet is probably especially important, yeah, for elderly people.
Okay. Can you speak to the benefits or side effects of supplements such as chlorophyll, echinacea, spirulina? Any evidence in regards to individuals diagnosed with MS or immune system?
I'm not aware of any research in that area.
Thank you. Can you comment on the use of extra virgin coconut oil? Does it taste good to you? Do you like it?
I like coconuts a lot, yeah. Especially if they contain a cocktail.
There's a whole other question then.
Yeah. I actually seriously don't know that much about coconut oil, but it's probably good. Yeah. But
It's saturated fat. The next question is, though, it is a saturated fat.
I really don't know that much about it. Yeah, I can't comment. Yeah.
Okay. Okay, here's a good one. Before I had my MS attack, I had good and regular bowels. Now I find myself with chronic constipation either because of the medication or MS. What can I do to reduce constipation and maintain regularity in my bowels? See, we do talk a lot poop today.
Poop is good. Yeah.
Poop is good. Poop is good.
Constipation, I mean, there are some of the strategies that I discussed today, such as dietary fiber. And there, depends on the fiber type, but there are definitely dietary fiber that are worth trying because they do increase gut motility and they might improve regularity, but it really depends on which fiber to use, so that's probably something that would have to be tried. And then there is some evidence that probiotics, but again, they are also not every probiotic will do this, so it's probably worth going into the internet, and there you have to be careful also, as I would definitely look into this chart with recommended products if there's something, but dietary fiber is probably what I would try the first. And then I can't specifically now say which dietary fiber, probably potentially, yeah.
Psyllium? Okay.
And then potentially also some of the short-chain prebiotics such as, false fructooligosaccharides or inulin, that could also be helpful.
Okay. Is there any benefit to regular fasting? Does it help to rebalance the human system and decrease the likelihood of common diseases?
I'm not aware of any evidence in that area.
The WALS protocol diet, are you aware of it? And what about the clinical trials and any comments on it?
I only read a little bit about the diet, and I'm not aware of any clinical trials.
Let me just read this one.
They are difficult questions.
They are. Oh, here's a good one. What is the average age of death for an Imiridium and a New Guinea study participants? Are they dying before the onset of chronic disease that might naturally occur in later life?
That's a very good question. So, but no, the answer to this is definitely no. So their average life expectancy is probably around 10 years less than you have in an industrialized society. However, much of this is driven due to infant mortality. So due to infections, they have I don't know exact number, but 20-30%, of individuals don't survive until the age of 5. And that really reduces, of course, the life expectancy dramatically. But there are clearly a large part of the population who makes it into 60, 70. So I think it is fair to say, and this has been studied, for many decades, that this increase in chronic diseases is not just that there's strong data of colon cancer, for example, which is much, much lower in these societies. So this is not just because, they die before these diseases become relevant.
Okay. You mentioned the PIA diet. What does the P stand for?
Did I mention that?
I don't know. PIA diet, autoimmune diet or something like that.
It might be on the slide. So these are actually diets that are on the MS side. I don't know. I read all of these diets.
I'll look
and
it up
what was
quickly.
striking for me is, that they often agreed on, their use on animal fat and plant material and fiber, but I don't know any specifics
I'll
about
confirm
that
what
diet.
that was.
Yeah.
Paleo diet, maybe that was
Oh,
it.
the paleo diet.
Maybe it was the paleo diet.
Okay.
Thank you.
So the paleo diet is a real mixed bag. Yeah. So because I don't think anyone knows what a paleo diet really is. So the idea of paleo is, that it's the diet with which humans have evolved over millions of years. And I did discuss this diet with some of the real experts in the area, which actually do archaeological studies, to really try to find out what was actually the evolutionary diet. So first, often the paleo diet, and by no means always, but often the paleo diet is rich in meats. And the experts that I actually talked with don't think that meat was that central for human diet throughout evolution. Now, clearly humans ate meat, but it was not the dominant diet. The dominant diet of humans, of hunter-gatherers, were actually plants and berries and nuts and plant products. Yeah. So as it's very hard to say what a paleo diet is, but when it comes to the idea of trying to eat more like our ancestors, I do actually think it is probably safer to say to go more towards plants, root vegetables, and definitely an increase in dietary fiber and probably in a reduction of meat.
So is there a test to evaluate your leaky gut?
Yeah, there is, and this is used in research quite a bit. So there are many. Also, I don't know if this is available, through general practitioning, but in research we have different tests by which we study barrier function or barrier dysfunctions, and this can be done by different markers in the blood of proteins that keep the epithelial barrier together, or it can also be directly measured by giving individuals a mix of sugar alcohols that they consume, and then you actually detect them in the urine, and the more of these sugar alcohols that make it into, am I pronouncing urine wrong?
Urine?
Urine,
You're
so
talking
obviously it is wrong.
Pee
Yeah.
now.
Pee, yeah. So it's sugar alcohols that are consumed, and then you measure by which degree they actually make it into the pee.
Okay.
And by this you can actually then make inferences on barrier function, yeah, in the gut.
Curious about the impact of lectins on the gut.
I
So
get-
also gluten and casein, but we already talked about gluten.
Yeah, I get this question every single time. There's a lot in the internet. You can't believe most what you find in the internet actually. And I discussed this with my nutrition colleagues at the University of Alberta. There is no really believable scientific evidence that a lectin diet is really detrimental.
Okay. Eggs, any comment on eggs?
So eggs are really interesting because if you look also from a nutritional standpoint, because if you, so it's a famous slide actually that nutritionists often use to show how unreliable nutrition research is. Also there were 4 cover of Time magazine within the last 50 years which all told you something completely different about eggs. Yeah. So also from, the greatest thing in the world to the worst thing in the world. Yeah.
Oh.
So the current standpoint is actually that eggs are probably not that bad. Yeah. So they're,
Well, I'm sure our egg farmers of Ontario would be very happy to
Yeah.
hear that. Okay, I'm just trying to, we've got very little time now, so we've got a lot of questions I'm trying to tie together. If I was to move to New Guinea and manage to survive without getting, killed by some animal of some sort, how long would it take for my gut microbe to begin to behave like a native?
That's a really good question. So I'm actually in the early plans to do some of these studies, also to think about, can we, bring people there and then study their microbiome. There are some smaller studies, which looked at this. It's really, more research has to be done. It's not sure if you can actually, acquire some of this diversity. There's some evidence, that it might have to happen early in life. Also meaning, if you don't get this early seeding of the microbes, that it's at least different. But then there's anecdotal evidence from, so there's a scientist in London, Tim Spector, and he wrote a blog where he traveled to the Hadza in Tanzania, lived there for a while, went back to London, sequenced the poop sample, and he actually had a higher diversity in his gut, and he lost it within 2 weeks. Also but again, we need systematic studies to really look if we can, replenish the gut by traveling to these places. Yeah.
What is your recommendation of how much fiber we should eat a day, or the
So
charts?
the recommendation is to eat probably as much as possible, but there is, I definitely think that recommendations from nutritional agencies are too low. So the recommendations are that, it's something like between 25 and 35 gram, or 28 and 38 gram, depends on which agency. And the amounts that are probably necessary are probably higher, and there are different colleagues who argue and for higher amounts. And a level that I hear a lot, which seems to be consistent, is that you need around 55 grams.
Okay.
Yeah. But again, there's also, it's very hard to do this research. Also, you have to take this. Also this is, but there are different scientists I know that have actually made the argument. For example, Stephen O'Cleave, who worked on diet in rural Africa, he had a diet with 55 grams. And then I've seen scientists who study on short-chain fatty acids and their physiological relevant levels. And I asked the question also, and again, I got the value of 55 gram. Yeah. So that's probably a value. At least that's a value that I target at the moment, also with our research.
And did I hear you say prebiotics are the vegetables and the fruit?
So, no, prebiotics are non-digestible carbohydrates.
Okay, sorry. So are prebiotics then just as important as probiotics, and what form then is prebiotics? Thank you.
So prebiotics it's a term that is used to describe some sorts of non-digestible carbohydrates. Basically, the non-digestible carbohydrates that are specifically designed to support good gut bacteria. I don't really see that much point in differentiating prebiotics from dietary fiber. For me, they are to some degree doing the same things in the gut. They provide growth substrates, and both of them are good. Yeah. So now you have possibility to get dietary fiber and prebiotics through the form of pills or supplements. And again, there's unfortunately not much studies out there if it's beneficial in multiple sclerosis. One thing I actually forgot to mention, some dietary fiber supplements have clearly been shown to improve cholesterol levels. And one thing that I learned at this very nice conference is actually from the previous two speakers is that cholesterol levels are really linked to multiple sclerosis, and that even statins are now discussed to potentially be beneficial. So there's definitely dietary fiber supplements by which you can improve your cholesterol levels. They are out there, and that research actually is clearly there. And then in terms, you can use this as supplements, but of course these compounds are also in diet, in normal food products, and there it's again vegetables, nuts, fruits, and whole grains. Exactly. Thank you.
The only transplantation you spoke about was the fecal transplantation. You cannot transplant one person's gut to another person's gut, so it's only through the fecal.
I mean, you can.
If you're dead, maybe, no?
No, my gut transplantations have been done.
Really?
Oh yeah, small bowel transplantations. In certain individuals, but
so what do you see? Would this be a possible treatment for people with MS?
Well, these are really hard questions. Yeah. So the bowel transplantations that I'm aware of are, they are due to illnesses, that affect this organ. Specifically, if you have a short bowel in children, you can make a small bowel transplant. I would think that. I can't really see how that would be a valuable treatment option for multiple sclerosis.
So I'm going to miss questions that Jens already spoke to, and you'll see in his slides when I send it to you, so you can be revisiting. So things like how to rebuild up your gut health through diet, etc. But there was a question about zonulin, Z-O-N-U-L-I-N.
Yeah, zonulin is a tight junction protein. That basically holds the epithelial cells together. Yeah, so these proteins can be supported through short-chain fatty acids, and short-chain fatty acids are the metabolic end products of fiber fermentation. It always goes back to fiber.
Okay. And somebody spoke about if you had a stem cell transplant, tell us how the microbiota could be affected after a series of chemotherapies. And that would be for anybody going through chemotherapy in general.
So yeah, this is one of the areas that is very interesting in the microbiome field. So there's very strong evidence in the field that with a stem cell transplant that the microbiome is actually severely disrupted, and that you often get even a very, very strong bloom of certain pathogens such as Enterococcus, for example, and then often these bacteria cause actually complications then further downstream. Yeah? So this is, there's a limited amount of research, but the research is very strong, showing that yes, you induce a strong alteration, specifically also because these patients receive, a lot of antibiotics, and what scientists now start to look at is to actually do fecal transplants from the same individual. It's amazing if a patient undergoes these treatments to actually collect poop samples, and then, you undergo the stem cell transplant, and then you put the same poop sample back into these individuals. But I think these studies are really just starting, to my knowledge.
Any connections to the gut biome and the appendix removal? And I'm also going to throw in there gallbladder removal.
Okay, the appendix, the hypothesis that is out there at the moment is that it serves as a reservoir for gut microbes. I'm not sure how strong the evidence is, but that is that's the idea at the moment. Meaning that, the appendix, because it collects a certain amount of bacteria, that this would then, if you take antibiotics, that you would be able, with an appendix, you would be able to, more easily rebounce, and the gut microbiome would be more easily re-established. I am not aware of the strongest, actually direct evidence for that, but that idea is being, thrown around, and it makes sense, yeah, but I don't think it has been systematically studied. And then bowel, as you said.
Oh, gallbladder, I don't know.
Gallbladder probably has very little to do with the microbiome. Yeah.
And you spoke a lot about the diet aspect, and the expense of diet. So here's the reality: organic over non-organic, GMOs over farm fish, etc. So, are we, if we walk into our local No Frills, are we getting the best products, or do we need to look at non-GMO and organic?
Man, everything what I talked about is basically, a plant, a high fiber plant, whole grain, fruit diet. So I'm not actually, at least in my own research, I don't really look a lot at organic and non-organic and gluten-free. I think it's really if I look at the modern diet, and I don't want to bring the table up again, but I don't think that these are the major thing. The major aspects of our diet is really, it is too rich in sugar, by far too rich in sugar, and it is by far too rich in saturated and trans fats, and it's specifically saturated fats, and it doesn't have enough dietary fiber. So I would rate these dietary factor higher than now necessarily, if this is organic or non-organic.
Okay, I am not aware what the low FODMAP diet is, FODMAP diet, yeah. But still having some gastrointestinal issues, even though following this after having severe C. diff colitis. Will it ever get better?
So FODMAP diet is a diet which actually tries to remove short-chain, non-digestible carbohydrates from the diet. Because as much as I talked a lot about dietary fiber and how beneficial it is, but of course it reaches the colon, it gets fermented, and in certain individuals, it potentially causes then, unpleasant symptoms and problems such as irritable bowel syndrome. So there is the idea, that by removing at least the carbohydrates that can be easily fermented, that this FODMAP diet is actually beneficial to prevent IBS. I do have to say this is still to some degree controversial, and I do really know some of the primary researchers that work in this area. And from a basic understanding of the fermentation processes in